Welcome to our channel Mootechie. This presentation covers Bergey's Manual of Systematic Bacteriology. In this presentation, we will discuss its history and editions, the manual structure, major bacterial groups, and its role in identification. This first volume focuses on the archaea and the deeply branching and phototrophic bacteria. Let's begin with an overview of Bergey's manual. It is the authoritative taxonomy reference, considered the global gold standard for the classification and identification of prokaryotes, which include both archaea and bacteria. This means it's the definitive source scientists turn to when naming and categorizing microorganisms. It is built by expert consensus, published by the Bergeys Manual Trust, and authored by over 1,000 international subject matter experts, ensuring its reliability and comprehensiveness. Its organization is phylogenetic, meaning it organizes taxa based on evolutionary relationships, primarily using 16S rRNA sequences rather than just observable traits. This provides a more natural family tree-like structure. It is a systematic manual which complements the older determinative manual that focuses on practical identification keys. The systematic version provides detailed descriptions, ecology, and information on type strains. It has broad application, being an essential tool for researchers, clinical diagnosticians, and environmental microbiologists worldwide. The image here represents the diverse prokaryotic morphology, illustrating the incredible variety of shapes like rods, spheres, and spirals, and arrangements that bacteria and archaea can have, which is one of the many features used in their description. Now, let's look at the history and editions through a timeline of taxonomy. In 1923, the first edition, titled Bergey's Manual of Determinative Bacteriology, was published. This initial version focused heavily on practical identification keys to help microbiologists name unknown bacteria based on their observable features. Then, from 1984 to 1989, the first systematic edition, Volume 1, was released, expanding to a 4-volume set. This marked a significant shift in focus towards understanding bacterial relationships and phylogeny, not just identifying them. The second systematic edition, from 2001 to 2012, comprised 5 volumes and involved about 1,000 authors. A key feature of this edition was that classification became largely based on 16S rRNA gene sequencing, a molecular technique. This leads us to the paradigm shift in methodology, which was the transition from relying purely on phenotypic traits like shape and staining to incorporating molecular methods such as 16S rRNA analysis and whole genome sequencing, or WGS. Finally, from 2015 onwards, the manual moved to digital format known as BMSAB Online, published by Wiley. This allows for continuous real-time updates to taxonomy as new discoveries are made, keeping the manual current in the fast-moving field of microbiology. The timeline shows the evolution from a determinative focus to a systematic focus and finally into the molecular era. Next, we'll examine the organization and scope of the manual. It is divided into a 5-volume organization, with each volume dedicated to major phylogenetic groups. This structure helps users navigate the vast diversity of prokaryotes in a logical way. The core of its taxonomy is phylogenetic classification, built primarily on 16S rRNA sequences and core genome analysis, which reveals evolutionary lineages. Each entry is detailed, including diagnostic traits, physiology, ecology, nomenclature history, and type strains, providing a complete picture of each organism. Importantly, phenotypic data is retained. Despite the strong genomic focus, traditional descriptions of physical and biochemical traits are kept to aid in practical lab identification. The manual also features extensive cross-referencing with keys, synonyms, and amended descriptions to help users find information. The volumes are Volume 1, covering the archaea and phototrophs; Volume 2, covering the Proteobacteria, which is so large it split into parts like alpha, beta, and gamma; Volume 3, covering the Firmicutes, which are low G plus gram-positive bacteria; Volume 4 covering groups like the Bacteroidetes, Spirochetes, and Tenericutes, and Volume 5 covering the Actinobacteria, which are high G plus gram-positive bacteria. Now, let's discuss some of the major bacterial and archaeal groups covered. First, the domain Archaea. These are distinct from bacteria, one key difference being they possess ether-linked lipids in their membrane systems instead of ester-linked lipids found in bacteria. They include fascinating organisms like methanogens and many extremophiles such as thermophiles that love heat and halophiles that thrive in salt. They also have unique 16S rRNA sequences and lack peptidoglycan in their cell walls. Next is the phylum Proteobacteria. This is the largest and most diverse bacterial lineage, and all members are gram-negative. It's divided into classes named with Greek letters like alpha, beta, gamma, delta, and epsilon. This group includes famous model organisms like E. coli and many major human pathogens. Then we have the phylum Firmicutes. These are mostly gram-positive bacteria with low G plus C DNA content. A notable feature is that many can form resistant endospores like Bacillus and Clostridium, which allows them to survive harsh conditions. This phylum also includes lactic acid bacteria, which are crucial for food fermentations like yogurt and cheese production. Finally, the phylum Actinobacteria. These are gram-positive bacteria with high G plus C DNA content. They often exhibit a unique filamentous branching growth pattern that can resemble fungal mycelium. Perhaps most famously, they are a major source of antibiotics, with the genus Streptomyces being a prime example. Now, let's discuss the applications and utility of Bergey's manual in bacterial identification. First, it provides standardized taxonomy. This offers a universal language and nomenclature, ensuring that scientists worldwide describe and refer to organisms consistently, which is fundamental for clear communication. It guides the diagnostic workflow. The manual helps microbiologists transition from initial phenotypic observations made under the microscope to more targeted and confirmatory molecular tests like 16S rRNA sequencing or MALDI-TOF mass spectrometry. It is critical for clinical stewardship. Accurate identification of a bacterial pathogen is the first and most crucial step in diagnosing infections, determining the correct antibiotic susceptibility, and ultimately ensuring proper patient care. It serves as a definitive reference. The manual offers comprehensive descriptions of type strains, which serve as the officially recognized reference specimens for a species. These serve as the ultimate benchmark for quality control in laboratories. Finally, it has vital industrial applications. Identifying specific bacterial strains is essential in fields like biotechnology for enzyme production, in food safety to monitor fermentation processes, and in bioremediation to select organisms that can clean up pollutants. The image represents the step-by-step laboratory identification process. Thank you. We hope this overview of Bergey's manual aids your microbiological studies. Enjoyed this presentation? Subscribe to our channel Mootechie. Leave a comment if you have questions. The primary sources and references include the Bergey's Manual Trust, the BMSAB online platform from Wiley, and standard microbiology textbooks. We hope this serves as a useful foundation for your exploration of the microbial world. Happy studying!